This talk will cover the basic patterns of Mendelian inheritance. The first major pattern is autosomal recessive. In an autosomal recessive trait, the phenotype is only seen in a homozygous individual, that is, an individual who has two copies of the mutant gene, one on each chromosome. Heterozygous individuals, and of course homozygous for the wild type, are unaffected. One typically sees affected individuals within a sibship. Both parents will be carriers and face a 1 in 4 chance of transmitting the recessive allele to a child who will be affected. One sees an increased frequency of consanguinity in instances of rare autosomal recessive traits. This is because an individual may become homozygous because of inheritance of a rare trait from a common grandparent. In this case, this common grandfather transmitted the allele A, little a, to his daughter, then to her son, and then to this child, and then in addition to his son, who in turn transmitted it to his daughter, and then to this child once again. The concept of autosomal recessive inheritance in humans was formulated by Archibald Garrod in the early years of the 20th century, just after Mendel's laws were rediscovered. He described a set of disorders that he referred to as inborn errors of metabolism that displayed this Mendelian recessive inheritance. Many, though not all, recessive traits are inborn errors of metabolism, where a block in a particular biochemical pathway, for example, in the conversion of phenylalanine to tyrosine, catalyzed by the enzyme phenylalanine hydroxylase, where a block occurs, for example, in this case, characterized by a phenylalanine hydroxylase mutation leading to phenylketonuria, or PKU. The concept, then, of a metabolic pathway is that substrate A is acted on by an enzyme converting it to substrate B, which in turn is acted on to convert to C, then to D, and so forth. A mutation in a gene, for example, gene C, will lead to lack of enzyme C, and hence a block in the conversion of C to D, leading to an accumulation of A, B, and C, and a deficiency of D and E. The phenotype can be any combination of accumulation or deficiency. Why is an autosomal recessive transmitted as a recessive? Well, in the case of inborn errors of metabolism, it comes back to the concept that enzymes are catalytic. So in a homozygous normal individual, if the triangles represent enzyme and these rectangles the substrate, the enzyme binds to the substrate, cleaves it, and then enzyme is regenerated. In a heterozygous individual, about half the normal amount of enzyme is created, and this is still sufficient to carry out the biochemical reaction, again because the enzyme is regenerated. Only a homozygous individual who produces essentially no functional enzyme will actually show the phenotype because of an inability to cleave the substrate. The second major pattern of Mendelian inheritance is autosomal dominant. Here, the phenotype is expressed either in homozygous individuals or in heterozygous individuals, and technically in a dominant one can't tell the difference between them. In fact, for many human autosomal dominant traits of medical interest, the homozygous affecteds, first of all, are exceedingly rare because the alleles will be rare, and secondly, oftentimes, the phenotypes in fact are more severe in the homozygous individuals as compared to the heterozygotes. By far the most common scenario for an autosomal dominant will be heterozygous individuals being the primary ones that one will encounter clinically. One sees in the pedigree vertical transmission from parent to child. Any affected individual has a 50% chance of transmitting the trait to any offspring. It doesn't matter if the parent is male or female, or if the offspring is male or female. So on average, as one looks at a pedigree, one expects to see about 50% of the at-risk individuals having inherited the genetic trait. Two concepts that arise particularly in the context of dominant inheritance are penetrance and expressivity. Penetrance is defined as the fraction of individuals who carry a gene who actually will manifest a specified phenotype. So one can infer, for example, that this individual is non-penetrant because she must have inherited the mutation from her father because she transmitted it to her daughter, and yet she doesn't manifest an apparent phenotype. Or here you notice a deficiency of affected individuals in this youngest generation. There would have been 4 children at risk, and none of them show signs of the disorder. Now, obviously, in determining penetrance, one needs to define exactly how the phenotype is searched for. If, for example, one means that a phenotype is looked at in a relatively casual way, like examining somebody fully clothed or from across the room, there may well be individuals who, if you look very carefully, would actually have the phenotype who might be missed on casual examination. Sometimes it requires blood testing or, for example, an X-ray in order to determine if a person really does carry the phenotype associated with a particular trait. So one has to be careful in terms of defining what we really mean by a person who's non-penetrant. One way or another, though, penetrance is an all or none phenomenon for a particular individual. However you define the phenotype, they either will express it or they won't. There's really no such thing as a partially penetrant individual, although one can specify a proportion of penetrance, which means, as defined here, that fraction of individuals who actually show the phenotype among those who carry the gene. Now, a special case of incomplete penetrance is age-dependent penetrance, in which the probability of expressing a phenotype increases with age. So here you see essentially nobody would be affected at birth, but the older you are, the greater the chance that you'll express the phenotype. And at least in this curve, if you reach age 90 and carry the gene, you're essentially certain to ultimately have the phenotype. A well-known example of age-dependent penetrance is Huntington disease, where very few children are affected, but if you have inherited the gene and live long enough, you probably will develop the phenotype. In contrast, expressivity refers to the degree or range of phenotypic expression of a trait. There could be individuals who are mildly affected or individuals that are severely affected, and there may be a bell-shaped curve of degree of expression. One could imagine an example of a trait, for example, extreme short stature, in which there is normally a bell-shaped curve in the general population of height. Some people are short, some people are tall. And in the trait, there's also a bell-shaped curve. Some people are exceedingly short, some people not so short, and there could be an area of overlap. We might define a penetrance as individuals who fall, say, to the left of this line will be defined as being penetrant for the trait, and those to the right are defined as being non-penetrant for the trait. Sometimes a recessive trait can be transmitted in a way that resembles a dominant. This occurs if the mutation is a relatively common one in the population. In that case, it will not be unusual for a homozygous affected individual to have a partner who is heterozygous. The homozygous individual will always transmit the recessive allele to each offspring, and the heterozygous individual has a 50% chance of transmitting the mutation to an offspring or not. So in this case, it appears that transmission is vertical, but in fact the trait is recessive. Again, this occurs if the mutation is relatively common in the population, an example of which would be hemochromatosis. The third major pattern of Mendelian transmission is X-linkage. Remember that males have a Y chromosome, whereas females have 2 X's. So if a male has a mutation on an X chromosome, he will be affected, whereas for a female to be affected, if it's a recessive, both copies of the gene need to be mutated. And this is bound to be a much rarer situation than the what we call hemizygous male. So the hallmark of an X-linked recessive trait is that it tends to be expressed more often in males, where females may be carriers who can either transmit the trait to a son or carrier status to a daughter, but it will be much less common for females to be affected. Most importantly, there is never male-to-male transmission because a male does not transmit an X chromosome to his son. If we're dealing with a dominant, all daughters of an affected male will get the gene, and female carriers transmit the gene to about half their offspring, as I've said. Indeed, there are X-linked dominants where a female or a male can show the phenotype. Here, half the offspring of a female are affected, all of the daughters, but none of the sons of an affected male are affected. There is also a special case of X-linked dominant in which the males have such a severe phenotype as to be miscarried in early pregnancy. Incontinentia pigmenti is a good example of this. So an affected female, if she transmits the trait to a male, that male will miscarry in early pregnancy. Any male who is born is bound not to be affected, and she transmits the trait to half of her daughters. Remember that during early development, one of the two X chromosomes will be inactivated at random in different cells. If the inactive X is shown in blue here, then either the maternal or the paternal X can be inactivated, but once it's inactivated, it will remain so in all cells that derive from that original cell. And incontinentia pigmenti is a dramatic example of this in an affected female, where you get these streaks of hyperpigmentation. They represent clones of cells either that express the mutation-bearing X and produce this darkly pigmented skin, or in the X that does not carry the mutation, in which case they get the lightly pigmented skin. I should point out that not all areas on the X chromosome are subject to X inactivation. In particular, the so-called pseudo-autosomal regions at the ends of the short arm and of the long arm escape X inactivation, and there are several genes along the length of the X chromosome that also escape X inactivation.